Hi, my name is Jeff Mentch, and I'll be talking about identifying brain networks in a clinically rich and naturalistic dataset using tensor decomposition. So first, I want to give some motivation about what we mean by a naturalistic dataset. So in this case, we're talking about people while being scanned with fMRI while they're watching movies. And there are a few reasons why we're really interested in using these sorts of stimuli. So the first is that they have increased ecological validity. So these conditions are thought to be more like the real world, at least compared to something like resting state or looking at some kind of sparse task design. Second, they're also very rich in these semantic, social, and perceptual features. And this is actually really important because we're interested in studying things like autism. And we know that in autism, people have altered social communication or altered perception. So these really rich movie stimuli might actually be a great way to look at some of these clinical conditions. So third, they're very engaging and they've shown to be useful in young and clinical populations. And they're also amenable to data-driven approaches, which we're interested in using. So now I want to give a little bit of motivation for our methods here and why we're interested in tensor decomposition, which is another buzzword in the title. So a lot of these data-driven decomposition approaches, like think of PCA, ICA, CCA, these are all pretty prevalent in neuroimaging research, and they're used, for parcellations or to find different networks. But many of them require certain assumptions. So if you think of something like group ICA, that has assumptions like all of your components that you end up with need to be spatially statistically independent. And that's actually a problem because these assumptions are not always necessarily biologically plausible. So for example, if you're watching a movie, you're getting these streams of auditory stimulation, you're getting all these visual features coming in, you're thinking about the plot, the characters, and the different parts of your brain are all dealing with this somehow, and it's very overlapping. So this is where tensor decomposition comes in. So tensor decomposition allows for the identification of temporally and spatially overlapping networks. And it's actually been successfully applied in fMRI data on resting state and in some clinical data sets, namely looking at ADHD. So now you might be wondering, what exactly is tensor decomposition? You may have not heard of it before. Basically, it is a method to find low-rank representations of a higher dimensional tensor, where a tensor here can be approximated by a sum of rank 1 components, right? So in practice, if we're thinking about our fMRI data, and we have one subject and one run, our data you can think of it as, it's a time course over our brain space. In this case, it's gray ordinates because we're working with SIFTY data, but that's one subject, one run. And there's different ways you can stack this. Like say you're doing spatial ICA, you could stack it in one dimension, or temporal ICA, you could do it in a different dimension. But instead of stacking it in a two-dimensional matrix, we're actually going to make it 3 dimensions and stack our subjects in this third dimension. So we end up with this third order tensor here. And then we apply our CP tensor decomposition, and we end up with n rank 1 components. And for each of those components, what you get is basically a temporal mode, which is like a time course for the component, a spatial mode, which is like a map of the brain of that component, and a subject mode, which is like the relative levels of the contributions for all the subjects for that component. And I do want to mention that the specific flavor of tensor decomposition that we're using here is called NASCAR, and that is Nadam Accelerated Scalable and Robust CP Decomposition. And this is just a robust way of solving the problem. And this was done by Lee et al. 2023. That's Andrew, who's one of the authors of this work here. So what are our hypotheses? So first, we know that NASCAR was previously applied to resting state data, but it's never been applied to movie viewing data before. So what we're going to do is do that for the first time. And our prediction here is that we'll be able to observe the same, or at least very consistent, spatiotemporal components compared to resting state. Second, by applying NASCAR to this movie viewing data, we're hoping to map the temporal modes of the components to movie features that we can extract from the movie stimulus to better understand brain differences in clinical populations like autism. And third, we want to classify autism diagnostic status from these subject level contributions, which was that third mode of the decomposition on the last slide. Okay, so into the methods now. So the data set we're working with is the Healthy Brain Network data set from the Child Mind Institute. And in particular, for the purposes of this talk, we're just looking at a small subset from site 1, and it's about 200 subjects, half of which are typically developing controls, and the other half are individuals with autism. And we're working with the fMRI data, specifically 2 5-minute resting state scans and 2 movies. One, The Present, which is 4 minutes, and a 10-minute excerpt of Despicable Me, which is 10 minutes. And the TR here is 0.8 seconds. And if you want more details about these runs, definitely check out the paper by Alexander et al. 2017. It's a wonderful data set. Okay, so for the fMRI processing, we're running fMRI prep version 23, and all of our data is SIFT-T2. So we're looking at cortical surfaces and subcortical volumes. We remove all of the standard confounds, detrend. We do a minimal 2 millimeter smoothing. We do some standard scaling. And then we actually do this group brain sync step, which is for temporal synchronization. So this is actually really important for the resting state runs, because movie runs are time locked to the stimulus, right? Everyone is viewing the same movie, but with resting state, you don't know what people are thinking about. So what this step does is it synchronizes everyone critically for resting state, but it also helps the movie performance too. And then finally, we run NASCAR, the Nadam Accelerated Scalable Robust CP decomposition, and we get our components. So now I'll get into the results and show you the components. And what we'll do first is we'll look at the spatial modes. So I'll just throw all of them up here. This was from the movie Despicable Me. Again, that was the 10-minute movie, and we ended up with 19 different components. And what I'm showing you here are these spatial modes of the components. And you can see some familiar looking networks, maybe visual or default mode, somatomotor. Definitely some familiar looking things, at least subjectively, are popping out. So the first step we had to do here, once we get our results, is sort of validate these, right? So I'll just show one example here. So from the same movie, this is the 17th component, and it looks like a pretty strong auditory specific network. But we can actually project this onto known parcellations and maps of the brain. So for example, we can project it to the Yo Atlas, or the Glasser multimodal parcellation here, or here the Cole-Antisevich network partition. And then we can kind of see, which most aligns with our component. And here you can see the auditory network is quite high, language network also high, or for the MMP, these sort of auditory areas are looking good. And we can actually rank these. So I'll now show you for the Cole-Antisevich network here, a ranking of all of the different parcels there. And we can see quite clearly that auditory and language are quite high, and the rest are quite low. So that does make sense, at least for this one example, component 17, it does seem to be a very auditory-like network. So that's great. Now we have a few questions. So the first question, are the same components observed across different resting state runs? So this has been shown by previous work from Andrew and quite strongly, but we want to just make sure that's still the case in this new data set. So just as an example, here's a visual network from resting state run one of the default mode network subsystem 2s from resting state run 1, and also a somatomotor network. Just as an example, let's now compare that to resting state run 2, and you can see the answer is yes, they're actually pretty closely correlated here, it looks like. And now our second question, are the same components observed across rest and movie viewing? So movie viewing has never been done before. We ran it through, and now let's take a look at the same networks, and the answer is yes. So you can look, you do see the visual and these same sort of networks. And I do want to add, these weren't manually matched. This was all done using the Gale-Shapley algorithm. And I couldn't fit all 17 on here, but definitely come check out our poster if you want to see all of them. Okay, so the spatial looks good. Now we can take a look at the temporal mode here. Okay, so now this is an example from the same movie, 19 components. Here's time, this is in TRs. These are all of the components. And with resting state, this isn't so interpretable, right? We've synchronized them all the subjects, but you don't really know what people are thinking about during resting state. So this is where the movies come in. So we know people were watching Despicable Me, at least we hope they were watching Despicable Me, and we can actually do some interesting things there. We can go to neuroscout.org, which is a great resource for naturalistic fMRI data and analyses. Definitely check it out. And there are all these pre-extracted features from all these great machine learning extractors. And we can just grab all of those for Despicable Me. We have 215 movie features that we have, again, over the same time here. And some examples of these would be, things like faces, different visual objects that you can see in the scene, if speech is happening or not, if music is happening, even low level. So these are low level, high level, auditory, visual. And we can now do a mapping between these. So we might be curious, which components are most correlated with which movie features? So we'll run that correlation. And now I'll show you the results of these correlations for that same sample network as before, component 17, which we thought was very auditory. Well, let's look at the temporal mode now. In black, this is the mean temporal mode of the component. And in the different colors, you can actually see the top 5 correlated features are all actually related to speech or features extracted from the transcript of the movie. So we do see speech and movie transcript related features are most correlated. So we're able to sort of validate these using the movies and the movie features pretty well. And then again, here's a bar plot. These are the top 25 features across all of the different components. And you can see it's quite selective for things like word count, audio set, speech, text length. Interestingly, music here is anti-correlated with these other ones, which is interesting. And if we look at the lower ones, furniture, light, too, these are actually visual features. So it's not very correlated with the visual sort of features, which makes sense. So we validated it again. Okay, so now we want to look at the subject contributions here, the subject mode. And what I've done here is I've just plotted for each of the components all of the subject contributions, and I've split it in blue. This is no diagnosis given. These are the controls. And in red, these are the people with autism. And you can see we did a t-test, and even after a very stringent Bonferroni correction, 3 of these are actually statistically significant. And in fact, there are more contributions from controls than individuals with autism, which is interesting. So what we can do now is we can actually map this back to the other modes. So we can see, oh, okay, interesting. These spatial modes of these networks had more contributions. And then next we can also map this back to the temporal modes and see if those also make sense. And we're in the process of doing this, and we can make an interesting story out of that. So in summary, so our conclusions. So yes, consistent components were identified across both movies and resting state, and they do correspond with known networks in the literature. Temporal modes for movie components can indeed be mapped back to movie features, as we've shown, and significant differences were observed between people with autism and controls in some components. And actually, if you just give all of those subject components to a classifier, we can actually get a classification accuracy here of around 60%, which is not too bad. It's definitely above chance, but that's not bad for a first pass. And some future directions. So the first thing is to expand the scope of the analysis to more subjects and sites. Again, we're just looking at just a tiny bit of the huge, wonderful HBN data set. So we can increase this, an order of magnitude and start looking at more things. And we also really want to implement a left out sample model. So, we did run this preliminary classifier and got some good accuracy, but really, we run tensor decomposition on all of the data, all of the people, and then we run a classification after the fact. That's actually sort of data sniffing. There's some data leakage. We should ideally implement this left out sample model. So that's quite important. We also want to run a regression. So we'd like to do a regression model and predict other clinical measures of interest, like the social responsiveness scale, which is available as part of the HBM, or hopefully if the ADOS becomes available, we could do that. So that would be great for the clinical side. So with that, I want to say thank you. Huge thank you to Andrew and Satra for all of our work together on this project. And I want to give an extra shout out to Andrew for making this great tool, NASCAR, developing it and putting all the code out. And it's actually quite usable and well documented. So if anyone wants to try out NASCAR, definitely check out his papers and his website for more. And if you're at the conference in person, do find us at poster number 589. We'd love to talk more and show you more of the components. Thank you.